Hey, what's going on? It's Mr. Salmingo.
And Mr. Xu.
We're going to be lecturing today about cell communication, which is chapter 11 in your AP Bio book, and hopefully understand how do cells talk to each other after this lesson. I'm going to go over some of the things. I'm going to go over the basics, and then Mr. Xu is going to help demonstrate and talk about the different things that we're talking about today. So first of all, cells, how they signal to each other. They can communicate by three different ways. So basically, like humans, we could talk, we could do body language, but cells have three ways. Animal cells have three ways that they communicate. Number one, they do direct contact. So we talked about gap junctions last time. So like here or here for plant cells, like they could talk like when they're right up against each other. They could also secrete things. So whether it's growth factors or neurotransmitters, they could secrete stuff to talk to other cells. Or they could do long distance, like, in your body from a different organ to another organ, they could do long distance through hormones. So here are some examples of the things that we're talking about. Here's local signaling. Here's what they call paracrine signaling, where a cell is going to secrete stuff and tell other cells a different, a different message. Synapse, which is in your brain. What happens is that electric signal is given from a nerve cell, and it diffuses across the synapse, and it hits a target cell in your brain. So that's local signaling. Long distance, we're talking about from your bloodstream from one part to another. Again, basically a cell will secrete something into the blood vessel, which travels all across your body and into another cell. The cell signals have three stages. Here are my three stages, and the whole lesson is about memorizing these three stages. Stage one, reception, another word for receiving. And it detects a signal molecule. This is a very important vocab word you need to know. It's called a ligand, and it's a signal molecule. It's the signal that it gets from one cell and given to another. Stage two, transduction, taking that signal and converting it into a form that could cause the cell to do something. And step three is the response. The cell does something in response to the signal that it gets. So picture-wise, here's step one, reception. So here you see this big purple protein. It's on the plasma membrane. And again, this signaling molecule is called a ligand. It's a special key that fits only in a special lock or a special protein, and that's called the receptor. That is reception, step one. Step two, transduction. Again, think of this as a fancy way of the game telephone. This protein tells one molecule, and that molecule tells another molecule. This molecule tells another molecule. That's called transduction. And then response. After it passes along the message, then the cell does something. So this whole lesson is just about these three steps, and we're going to go very, very slowly and very descriptively. Okay, so step one. So reception, first of all, again, as we talked about, it's from a ligand, and the receptor is very specific. It only takes a specific signal. It doesn't take every signal. There's two types of receptors. We've got the plasma membrane receptor, which is on the plasma membrane, and it accepts water-soluble ligands. There's intracellular receptors, like the cytoplasm or the nucleus, like, within a cell, and they're hydrophobic. They don't like water, or they're small ligands. This is examples are testosterone and nitric oxide. So when the ligand binds to receptor protein, when it binds to that purple thing, the protein will then change shape, and then that shape basically initiates a transduction signal. So that's all what reception is. But there are three types of receptors, and again, I'm going to introduce it, and then Mr. Xu is going to talk about the actual receptor. So 1, 2, and 3, here we go, and we're going to talk about each of them. So the first one we're going to talk about is called the G protein coupled receptor, or you call it the GPCR. So this is what it looks like. This is a GPCR. Again, we just look at it as a big purple thing, but this is what it looks like. It has a binding site, as you can see up here, and then it has a bottom part that interacts with what they call G proteins. And then if you want to look at it even more in depth, this is what it looks like molecularly, or like more in depth. Here's my phospholipids, and then here's my G protein coupled receptor. That's what it actually looks like. How it works is what Mr. Xu is going to tell you right now.
So basically, you guys can see these four pictures. And as you can see, this part is kind of like how everything's supposed to look like when there's no signal. So you can see, oh, right here, sorry, my bad. So the G protein, the little purple thing that we see from the other picture, it's also represented as purple right here. What happened is the specific key, the ligand comes in and then binds with the G protein. What it does is start going for the second G protein that's inactive, but once it binds, it will go for that one and then change that one into GDP. From GDP change into GTP. Once the second G protein is changed into GTP, that second G protein is going to go interact with the enzyme that's going to be floating around on our membrane. And what it does, it's going to activate the rest of the part. So the G protein does by two things. Okay, that's the reason why we call it G protein coupled receptor. the first one go couple with the second one go interact with the enzyme usually, and then that enzyme will just activate by changing shape, okay, like how Mr. Samingo was saying. And once that changes shape, it goes down into a different kind of cellular response into later life. And what happened after that is this actually doesn't stay on forever. This GTP will be basically use up energy, and then one phosphate will come out, and then this one goes back to the normal off stage.
So in short, basically the GPCR, we've got 7 transmembrane segments, so this GPCR has 7 parts. And again, as he said, the G protein plus the GTP, it activates the enzyme, it changes shape, and then the cell responds. The second one is called tyrosine kinase, and this is, again, something that Mr. Shi's going to talk about. Here's a diagram that he's going to explain.
Okay, tyrosine kinase basically talks about, well, we need the amino acid tyrosine in all the kinase right here. And what happened is, it's also a receptor that is that's embedded in the membrane. And what happens is you have 2 ligands, okay, and both ligands has to bind to the ligand binding site. So when they bind together, that time they basically come together, like clapping your hand. Excuse me. So once they bind together, what happens is it attracts ATP, and ATP will come and actually drop off 6 phosphate on this 6 tyrosine, okay? And then the 6 tyrosine will be also attracted by something, the 2 enzyme will be attracted by these 6 phosphate, and they will combine with them. Okay, and what happens is, again, it changes shape and it releases and it goes into cellular response. And then it usually will activate 2 different things. Sometimes it will activate the same thing, but that's very, very rare. But usually 2 different things. So you actually activate multiple cellular response with this mechanism. And just like the G protein couple receptors, this one also goes back to normal. So once this is finished binding, and then everything goes back to this resting state. And this doesn't stay on forever. Otherwise, your body would just do the same thing over and over again, and that's just not gonna happen.
So in short, or in summary, remember the GPCR we just saw? We had a protein, then it had the G protein, and it went over across to the other side and activated an enzyme. This one we have 2 pieces, and then at the end we had these P's that attach to the end. And what's great about this one, it activates multiple responses at once, where we just saw for GPCR we only saw 1. And then the last receptor is called ligand-gated ion channels.
Okay, ligand-gated ion channel, it's similar to what we talked about. Remember, we kind of talked about the sugar fingers on the cell outside. Basically, this is kind of like that, but you can imagine, like, if you have you can't see the door, but you can imagine there's a door right here. Okay, imagine in the library there's a door, and usually there's a door like closed, okay, and it's usually locked. Okay, so imagine all of you are inside this door, okay, inside this library. Okay, with the ligand coming in to open the door, it's like a key. So it comes in to open the door, and the door will open. And once the door opens, basically you guys go all rush out once the bell rings. So basically this is what it does. Okay, a key comes into a door to open the door. Once the door opens, everything goes through, okay, and then the door will just close, and then ligand just will pop out, okay. And this would basically everything is about shape, just like Mr. Samingo say. So this key opens the door because it changes shape of the protein, and once the key leaves, the shape of the protein changes again, go back to rest state. So it closes its door again. So this is kind of like how they control different ion going one way or another way. Sodium, yeah. Usually sodium, calcium.
So in short, what he was mentioning is that, again, the signal on the receptor, it changes shape, which allows the ions to flow in. You saw those green dots in the last slide. So the gate opens, green stuff comes in. And those ions, as he mentioned, calcium and sodium are the ones that tend to go in that way. So that, again, that's step 1. That's all receptors. Step 2 is called transduction. So in transduction, I'll give you the summary, and then again, Mr. Shi will explain more in detail. But we have, essentially, we receive the signal, and now I need to pass it along like telephone. So we have cascades, you may have heard the word cascades before, of molecular interactions that relay the signals from the receptors to the target molecules. There's something called the protein kinase, And this enzyme, it phosphorylates, which Mr. Xu will talk about, and it activates protein. So it activates something, which activates something, which activates something. That's the enzyme that's in the middle. That's like the telephone game. And then the phosphorylation cascade, they're the ones that take the signal, they enhance it and amplify it. So it just keeps going back and forth, passing along the signal. So it makes more sense if you see the diagram, which we'll show you. So just real quick, here are my protein kinases. They're inactive, and once it gets active, they call it the active protein kinase. And then I'll have Mr. Xu sort of tell you what goes on.
Okay, so basically, just like what Mr. Domingo said, the first part receptor got it, and then this activates the whole chain of reaction. And this is usually what all the magics work. Okay, once the medicine comes to bind in this part, sometimes it comes to bind on the surface membrane part, and it activates a whole bunch of proteins within it. And so what happens is it activates this one, and then it activates another one, and then you just go on so forth. And what Mr. Domingo said, very important part. Oh, sorry, my bad. The phosphorylation cascade. Okay, what it does, you can see right here. This one activates the protein. Let's say this is protein number 1, and protein number 1 goes to activate protein number 2. But you can see right here, this one will come back and becomes inactivation again. However, this one will stay activated. So this signal actually gets amplified as you go down the chain. So it can just basically you just need one little molecule to activate like a huge and to create like a huge army. So imagine you're like the general of this entire army, and you want them to move to, I don't know, from factory 1 to factory 2. All you need to do is make a phone call, and then all of them will move down. Okay, so basically it's a chain of command, and then this one will always go all the way in until the cellular response, and this won't stop until the DNA and RNA is some kind of protein or some kind of new molecule are made because of this cascade. And again, once everything happened, once everything is done, everything goes back to rest. So this chain would not be stopped until everything is go through. So once everything goes through, it all go back to normal state. This is not gonna go on forever. However, some of the disease is actually messed up on this part. So what happened is it will go on forever, and the cell would just do whatever it's not supposed to do for a long time, and then they'll just do that over and over again.
So then, just in addition to those first 3 that we were talking about, there's also something called second messengers. Second messengers are small, they're non-proteins, and they're molecules or ions that can relay signal inside a cell. So like a second messenger. So for example, we have something called a cyclic AMP or C-AMP, calcium ions, or a inositol triphosphate. So how it looks like, I don't know, do you know how these work, Mr. Xu?
So basically, it's the same thing, except remember right here, this picture, it shows you this is a molecule. The relaying molecule, sometimes it could be a protein as well. But in this case, the second messenger is usually some kind of molecule that's way smaller than large protein. Well, compared to large protein, they're very, very tiny. So what happened is they basically do the same thing. They goes into a different cascade, but it's not a protein cascade. Okay, so just keep in mind, second messengers are not really like a protein cascade member. They're just kind of like the letter itself that's delivered to different factories. Okay, instead of general sending a lieutenant or somebody, a captain or colonel to the factory and then say this is what is supposed to be done. So think about second messenger as the letter or some kind of message, and the protein cascade as like an actual person going into different part.
So here's one example, C-AMP right here, and here's another example, IP3 as another second messenger. So the last piece, remember we're talking about 3 steps, then there's the response. What does the cell do once it gets the signal? So a couple things that it can do. It can regulate protein synthesis. So remember, synthesis is to make, so it can regulate proteins being made by turning on or turning off genes in the nucleus, otherwise known as gene expression. Or it could regulate activity of proteins in the cytoplasm. So that's what the cell can do once it gets that signal. So the bad story is, what if this whole thing that we talked about, the first 3 steps, reception, transduction, and response, what happens if these pathways are blocked, or what if they're defective? What goes on? So a lot of stuff that could happen. Diabetes, cholera, autoimmune disease, cancer, neurotoxins, poisons, pesticides, drugs. So these are anesthetics, antihistamines, blood pressure medications. These are all things that affect the transduction pathway. And we're going to talk about some specific examples. Mr. Shu and I will talk about a different example of what happens when that pathway breaks down.
Or work.
Or work.
Yeah.
In a good way.
In a good way.
In a good way.
In a good way.
So first one, we'll talk about cholera. You may have heard of this disease before. Cholera is something that you get, not that you'd want to do this, but if you drank contaminated water, usually with human feces, then you contract a disease called cholera. This is what it looks like. And what happens is that this bacteria is called Vibrio cholerae. It colonizes in your small intestine, and it produces this toxin. That toxin, how it works or how it makes you sick, is that it modifies the G protein. Remember we talked about G proteins. The one that deals with salt and water secretion. So a cell that, the part of the cell that deals with salt and water, salt and water, salt and water. That toxin modifies that G protein. What happens to that G protein is that it's stuck in active form. So Mr. Xu was talking about how a protein goes from active to resting, going back and forth. In this particular case, it's stuck in active form. And if it's stuck in active form, the intestinal cells, they secrete salts, they secrete water because it's always in active form. It doesn't go back to rest. So because you have so much salt and water in your body, that means someone that's infected, they develop profuse diarrhea. They poop a lot. And they could die because their body loses so much water and so much salts, then they could die because they don't have all the water and salt. So that's one particular way that messing up that cell signal transduction pathway would mess someone up. Mr. Shi will talk about another example.
Viagra. So basically, it's the same thing. So instead of making it a bad thing like the other example, you have diarrhea and all that stuff. Basically, what happened is we modify some kind of molecule to bind with the CGAM. Okay, and what happened is we doesn't allow it to hydrolysis. Okay, we don't want hydrolysis to happen once we take this. And what happened is it's like what the other the other slide is saying, it's always active right here.
Oh, hi.
But the other one is actually talking about there are always. That's Mr. Jackson.
He's a loser.
Okay,
I
didn't say that. Okay, anyway, so it relaxes the smooth muscle. So everything it works is it increases blood flow. Okay, so it will always stay that way. So there's a lot of blood flowing to that because the cells are all doing exactly the same thing. And because the high concentration of this medicine throughout every single cell, so the blood flow will be high all the time.
What are you guys watching?
Nothing, just giving a lecture.
Apoptosis. Oh.
Mr. Jackson will lecture about.
Yeah.
Sounds pretty dark.
Apoptosis is cell suicide. That's dark. This is awful. Why are you teaching them this?
So apoptosis. Okay, apoptosis is something that you will have to learn, and basically, actually, this is the part that makes who you are. Okay, so basically, a really quick example of what it is. It's a signal for cells to notify itself or notify its neighbor that it needs to die. Okay, and what happened is some cells are made in such a way that they're actually not healthy. So once they're not healthy, they have to die. Otherwise, they're just gonna create something bad in later. And example of that is actually cancers. Cancer cells doesn't have this precautionary, so and cancer cell would just grow, and they just don't know when to die, so they just don't die. So what happens is you get cancer. Okay, another two examples are Parkinson's disease and Alzheimer's disease. Okay, so another good thing about apoptosis, it's my bad, right here. See here, cancers. So this is what happens. So basically, it tells the cells, normal cells right here, and not very normal cells right here. So basically, when it becomes non-normal cell, it needs to die. And basically, there are multiple ways for it to do it. For this one, it shrinks and then it becomes like a blob, and then a bigger white blood cell, a different type of white blood cell, and just kind of eats it up. Just like that. And then this is basically the positive things, okay? So what happened is when we are growing that time, our hand is actually grown this way. And imagine it's like I'm wearing like, I don't know, some kind of ball around it. Yeah, a mitten. Here we go, a mitten. So the entire hand is grown this way, okay? But once the cell says, you know what, this is like I'm a 3-month-old baby, I need to start growing some fingers or something like that. So what happened is the cell actually know from the top, the tips right here, and then the cells around here will say, okay, it's time for us to die. but in a good way.
In a good way.
So the cell dies, and it basically creates finger, as you can see right here. And then
Hey.
so does the same thing with your fingers. Your fingers created this way, your toes, and then your arms and all that stuff is created the same way. And so is your facial, like everybody looks different, and because of this reason, okay, it shapes, and then basically some part dies, some part grows faster, and then basically that's like a positive thing about apoptosis, and it's all because of this transduction. It works, and this cell and cell communication works. Okay, and if it doesn't work, just like what Mr. Samingo say, something goes wrong, stays active all the time, or stay inactive all the time. It's neither of them is good.
So that's our lecture. Hope you enjoyed. Thanks for watching.